Momita Das. She goes by Mo. She's from the Rochester Institute of Technology. She works on computational and theoretical modeling of filaments, networks, and network-like materials from filament structured tissues. And as I learned, she was also a student here in 2001, and she'll tell us a bit about that. Please.
All right. It's really a pleasure to be here. Thank you, all the organizers, and Leo for keeping this really nice summer school going, and for all the organizers for organizing this wonderful conference. So I was actually here my first year of grad school. When I was starting grad school, I was here at the 2001. So that I'm kind of dating myself, and I'm also dating Leo by doing this. So my first year of grad school, it was on non-equilibrium statistical physics in soft materials, biomaterials, and so on. And then, and I did my PhD at the Indian Institute of Science, Bangalore. So that was my first year. Then I did a postdoc at Harvard, another postdoc, which was split between UCLA and Free University in Amsterdam. And then I ended up staying much longer in Amsterdam because, Amsterdam, well, LA and Amsterdam, I don't have to tell you. I also had a Dutch fellowship. And now I'm a professor at the Rochester Institute of Technology. So it's kind of nice to sort of circle back and be back here now as a lecturer after having been here, 2 decades ago as a student. And I guess a lot of things have changed, but I guess, Leo, you are kind of the constant, and the school is the constant. So, because I missed, so I would have loved to come here on Sunday, but the reason I'm arriving late is because I was also at a different summer school. So many of you have heard about the physiology course in Woodsall, Massachusetts at the Marine Biology Lab. So I was doing a rotation there on mitochondrial networks. So anyway, so I work a lot on network-like systems. So I guess because I didn't introduce myself when everybody else did, I'm going to spend the first 5 or 6 minutes telling you a little bit about what I do, and then, getting into the topics that I would like to discuss with you in this summer school, right? So, okay. So we all know that cells and tissues are soft and squishy, right? They can easily change their shape and remodel themselves. But at the same time, they're also really robust and resilient, and they can easily adapt to different mechanical contexts. And one of the things I am really interested in understanding is what are the physical rules, laws, mechanisms that are responsible for these kinds of properties of cells and tissues. So you guys have probably all seen the first, or many of you have seen this first movie. So it's basically, it's the most viewed biophysics movie on YouTube, right? So you have a white blood cell, which is chasing a bacteria, and these are the bacteria, and it's these are red blood cells, pushing the red blood cells out of its way as it does so. And it's quite relentless, right? It's generating force and sort of in a very driven manner, chasing this bacteria. It's also sort of changing its shape and reorganizing itself, but it doesn't really lose its physical integrity, right? It's not falling apart, right? So it maintains that. So while it's soft and squishy, it's also quite robust and also quite resilient, right? It doesn't break or fall apart, right? And this is my other favorite example of a living system that is soft and squishy and at the same time very robust, as we all know, right? So I don't have to, belabor that. So a little bit about what, I study. It's basically why are cells and tissues squishy but robust and resilient? What are the physics laws and rules? And if we understand this, can we recapitulate these kinds of properties in sort of synthetic biomaterials, right? So that's what my sort of big picture, questions I'm after. And so the way we try to understand this is sort of model the physics of network-like materials in cells and tissues, right? And we'll talk about that in this lecture. So some of the systems that my group looks at the level of cells, we look at cytoskeletal networks, we look at networks of mitochondria, we also look at sort of, organization of sort of biopolymers in bacteria. And then at the level of tissues, we look at extracellular matrix networks in our cartilage tissue. We look at synthetic materials, which we can make by combining biopolymers with different types of circadian proteins. And then, make synthetic materials which have some tissue-like properties, right? So these are some examples of things that, I study. And then the core, basically, principles that are there in these systems, there is interplay of mechanics, statistical mechanics, and geometry or morphology. And by geometry and morphology, things that can come in are, many of these materials have sort of composite multi-component structure, right? There can be spatial heterogeneities, there can be anisotropy, and so on. And you can also have dynamics and activity and so on in these materials, right? And then what we do is we basically write down mathematical models, which could be either in terms of equations of motion or free energies and so on and so forth. And then the method is basically solving ordinary differential equations, partial differential equations, minimizing, energies or some cost functions. And then, we try to solve these things analytically when it's feasible, but real biological systems are often very, very messy. And often you have to then, use numerical simulations to sort of solve these kinds of things, right? And I also really work in very close collaboration with experimentalists, where we are doing these kinds of modelings sort of iteratively with, experimentally. We basically inform our models. And one of my experimental collaborators, Itai Cohen, is actually lecturing in the last week of this summer school. So I'm not sure if he's going to talk about our stuff or he's going to talk about some other things. But it's really nice and satisfying to do that as well, right? So this is a little bit about who I am and what I do and sort of my trajectory, my interests, etc. The other thing I wanted to mention is that as we have this discussion, today and the next 2 days, please feel free to interrupt me anytime with questions, right? I would really like you to interrupt me anytime you have questions. So the structure of my lectures will have roughly this kind of outline. So today, I want to, you guys have already heard about biopolymers, but I'm going to talk a little bit more about their networks, how we characterize the mechanics of their networks. I'll talk about rigidity and rigidity percolation, what that is. And then I will basically ease you into the theory of this, theories of rigidity percolation in networks by talking about spring networks, right? So that's the discussion we will have today. Tomorrow we will have, rigidity percolation, etc., but not of spring networks, but of semi-flexible networks, right? And we'll see what is the difference between springs versus semi-flexible filaments. Once again, some of these things you might have already heard in the last 2 days, but because I was missing, and I think it doesn't hurt to reiterate some of the things, even if you have heard it, right? So, and then application of these kinds of things to, mechanical transitions in vitro systems, which are basically purified extracts, right? So real tissues and cells are quite messy, and a lot of things are present at the same time. But experimentalists, in order to do more controlled experiments, sometimes build purified extracts of one type of biopolymers from cells and tissues, right? And study them in vitro. So I'm going to talk about applications of some of the studies that we are doing to in vitro, cytoskeletal and ECM networks. And the kinds of networks I'm going to talk about will have, some of them might be homogeneous, some of them might have spatial inhomogeneities and so on. And then the last day I want to talk about mechanics of networks that are multi-component or composites, and also that might have activity, right? And then applications to then, mechanical transitions, mechanics and mechanical transitions in cells and tissues, right? So basically just, we will increase the degree of messiness or complexity, over the 3 lectures, right? So that's kind of the plan. And so today I am mostly going to use I'm going to use the board only if absolutely necessary, because this is more simpler things, but the next 2 days I'm going to use the board. So that's also the plan for, All right, okay. So we saw that our cells, this white blood cell I was showing you, is really soft and squishy, right? And this is really important because let's say when, we get a cold and our, white blood cells have to fight infections, they have to really go to sites of the infection, right? So they sometimes have to go through really narrow regions, right? So they have to change their shapes. Sometimes they have to move faster, et cetera, et cetera. So all of these things are really important. So you can ask, where is the mechanics or this mechanical property, like squishiness and so on of cells coming from? And it's mainly due to the cytoskeleton of cells, right? So like we have our skeleton, right? Cells also have a skeleton, but their skeleton is much more deformable, right? And it can reorganize itself. Our skeleton is not really reorganizing itself like the skeletons of cell do, and they can also generate forces. And they look like the scaffolding made up of different types of biopolymers, right? So here you can see many cells, right? Here we are looking at just one cell, and you can see that there's this polymeric scaffolding which look like networks or mesh, right? Made up of different types of polymers, right? So the cytoskeleton of these cells, and I'm mainly focusing, I'm not thinking of plant cells, I'm thinking of sort of animal cells and eukaryotic cells, right? So there are three major types of biopolymers in our cells, right? So there is F-actin or filamentous actin, and that's in this image, it's in this purple stuff. There are intermediate filaments. An example that you might have heard of is vimentin. And then, there are microtubules, right? And microtubules are the more stiffer looking ones, the yellow ones here. And all of these biopolymers are semi-flexible, and they form network-like structures. These networks could be physically cross-linked networks via different types of cross-linkers. Sometimes they're entangled networks. So no physical cross-link, but steric sort of interactions, right? Do you guys all know what are semi-flexible biopolymers? I don't know if you already went through that so far. No. Okay. Okay. So we all like food. So this is my way of talking about semi-flexibility. Okay. So if you think of polymer-like materials and their stiffness, let's think about spaghetti. So when you're spaghetti, you have not cooked your spaghetti at all, right? It's completely uncooked and stiff. So stiff polymers are like rigid rods, right? And they would look, if they were, on the scale of a cell or something, right? They would look like rigid rods. Polymers that were completely flexible would look like your spaghetti that has been cooked very well, right? So it has, it's very, very floppy, very, very flexible. And if your spaghetti is basically just al dente, right? So those would be more like semi-flexible polymers, right? In terms of your mental picture of what is semi-flexibility. So we can try to pin that down a little bit, right? So, and the stiffness is progressively increasing as we are moving more and more towards, stiff polymers. Okay, so what this physically means is that for semi-flexible polymers, the energy cost of bending these polymers, we cannot really neglect that. It can be sort of comparable to the energy cost of stretching these polymers, right? So for polymers, always there is a stretching sort of energy, right? And it could be two types. It could be because it's all coiled up, right? Because of finite temperature. And as you stretch it, you are stretching out this coil. So that's more like entropic stretching, right? And then there could be enthalpic stretching where you're stretching against the backbone, right? So there is this stretching energy, but for completely flexible polymers, we can neglect the bending energy because it costs very, very little energy to bend. It's, very small compared to the stretching energy, but for semi-flexible polymers, you cannot neglect the bending energy anymore. And for rigid rods, it's actually quite it's even more. Yes, you had a question. Yeah, enthalpic stretching is basically, let's say if I have a rod or something, right? So it's not like there is no thermal like anything, right? And I'm just stretching, against the backbone, right? So it's just that basically not temperature dependent, right? So it's athermal stretching. Yeah. Okay, so often we write down the bending energy, right, of these polymers as 1/2 times this bending rigidity. And then it will depend on the curvature, right, how much it's bent. And this kappa is the bending rigidity. We often approximate it as the Young's modulus times the radius to the power 4. right? And then we often characterize bending of polymers by this quantity, which we call a persistence length, right? So what is a persistence length? So if you draw tangents like this, right, and this is the angle the tangent makes with this direction, if you look at their correlations, these correlations, depending on how much this polymer is bent, if it's coiled a little bit versus if the bends are very large, right, you can write, how these correlations will decay as this exponential, right? And this S and S prime are points on the contour of this thing, right? And this LP is the length scale with which these correlations decay. And it's known as the persistence length, right? It's basically, it gives you a length scale. It gives you an idea over, what length scale over which these polymers are bent, right? And if I wanted to, relate it to the bending rigidity and the temperature, right? So this persistence length is basically directly proportional to the bending rigidity and inversely proportional to temperature, right? So if you are looking at a system which is at very high temperature, it's going to be much more bent, right? And the persistence length, the length scale over which you see these bends or folds, it's going to be much, much smaller, right? So is the idea of persistence length clear to all of you? And why am I talking about this? Because different biopolymers have different persistence lengths, and this plays an important role in their mechanical properties. So let's say, for example, for most of them, their Young's modulus is of the order of 1 gigapascal, right? So 10 to the 9 pascals, right? But their radius, right, the cross-sectional radius could be really very different. So for actin, the cross-sectional radius is of the order of 3 nanometers. For microtubules, it's of the order of 10 nanometers. For DNA, it's of the order of 0.2 nanometers. So then I can use this, all of these things to sort of calculate the persistence length and this temperature, I'm assuming it to be like room temperature, right? So KBT, we know that at room temperature, KBT is around 4.2 piconewton nanometers, right? So that's a number that's good to remember. So then the persistence length of actin is of the order of 10 microns. For microtubules is of the order of 1 millimeters. For DNA is of the order of 100 nanometers, right? So now, think about the size of a cell, right? If you forget about neurons or cells like that, most cells, are going to be, let's say, I don't know, 100 microns or something like that, or maybe a little bit larger. Over that length scale, right, microtubules would look like rigid rods, mostly, because microtubule persistence length is 1 millimeter, which means that in order to see these folds or bends in these microtubules, those will appear over these kinds of 1 millimeter length scales. But cells are not this large. Cells are much smaller than this. So on the scale of a cell, microtubules will behave like rigid rods. For actin, on the other hand, the length of the filament is also of the order of 10 microns or 20 microns or something. So the contour length and the persistence length are of roughly the same scale for actin, right? So you will see some bends, and it's smaller than the cell scale. So for actin, you will see some bends. DNA, you will see many, many more, right? So this gives you an idea of how much, what these things look like and what is their bending versus stretching. Yes. That's sort of the rough Young's modulus, I would say. I mean, more, I would say, in compression, because the thing is, some of these things are nearly inextensible, right? So, but again, that's kind of an estimate, right? Not everything is 1 gigapascals, but roughly that's a good, it's a good Fermi estimate for these kinds of systems. And I guess any of you, Patricia or Marcus, feel free to correct me if you have, better estimates for some of the numbers that I'm talking about. But roughly, I think 1 gigapascal is okay for the Young's modulus. The main difference is coming from the radius of these filaments being very, very different. Okay. So this is what semi-flexible filaments are, or polymers are. Basically, you cannot neglect the stretching energy. You have to take into account, you cannot neglect the bending energy. you have to take into account both stretching energy and bending energy of these polymers, right? But now in cells and tissues, it's not just individual polymers, they also form networks, like I said. And these networks could either be cross-linked by physical cross-linkers, or they could be entangled. And some examples of networks that I'm showing you, here is a network of collagen, in vitro network of collagen, right, from our extracellular matrix. This is a network of actin cortex in the cytoskeleton of cells, network of vimentin, which is an intermediate filament network, and this is a network of actin and spectrin in red blood cells. And you can see this network-like structure clearly in all of these things, right? So this is just to give you an example that indeed these things do form networks. Yes. So most of these things I'm showing you are in vitro. So people are making, like, they are taking purified extracts. So for actin, let's say from rabbit or something else, you can order it, and then you can grow these polymers and their networks. They are often, it depends on the cell type. So, but in early 2D, like some of the things I'm showing you, many of these experiments were sort of quasi 2D. So there was a little bit in 3D, but then mostly 2D. But in actual cells, these could be quite three-dimensional things. Again, depends on the cell type. So if your cell is moving on a substrate, right, and adhering on a substrate, then it would be more like 2D because it's moving on a surface and it's stretching out, etc. Right? And it's pretty, I would say quasi 2D, 2D and a little bit more, right? But it could be 3D depending on the cell type, because if you have a cell that's moving in 3 bricks, then that would be more, the skeleton would be 3D. But if a cell is moving on a flat 2D surface, it depends on what is it adhering to. Is it just moving on individual fiber-like tracks, in which case it could still be 2D, or is it just moving on surfaces, right? Sorry? Yeah, I mean, I think he's asking overall thing, right? Like in terms of how should we, like, compare the aspect ratio and comparison of the dimension, right? So I think usually people think of them as 2D or quasi 2D, but it's not true that they will always be. Like in an actual cell, the cytoskeleton could be quite 3D. So if you look at, a fibroblast or a cancer cell that's moving through a collagen matrix, right? So the cytoskeleton of those cells could have a pretty, like, a z-direction, like, it would not just be that, oh, the 2 dimensions are really large and the 3rd dimension is very tiny. So you could control, you could make a 2D network, you could make a 3D network. I guess I'm not an experimentalist, but by confinement, I guess you could put it in between cover slips and make it more. Yeah, and yeah. You can coat your cover slips also with stuff so that, things will be more, 2D. Yes. You could, I'm going to go into that more. You could change different things of the polymers with drugs. So let's say, for example, the different types of crosslinks, etc. You could change. Different types of crosslinks will have different properties, but you can also digest away some of the stuff, right? So for some of the things we will talk about things as a function of density of polymers. And either you could start with different concentration, but you could also degrade things away by using different types of enzymatic, enzymes, etc. Yes. It depends on the system. So for these kinds of things, twisting might become important when you, let's say, have not just filaments, but let's say if you have a bundle of microtubules or something, then for certain time, so the best answer to your question is it depends on the context. And for fiber networks of filaments, it may not be so much. Even networks of filament bundles, it may not be so much, but individual bundles, etc., then, like microtubule bundles and so on, there, twisting might be important. All right. Okay, and now, so then you can ask, how do we characterize the mechanics or rigidity of these kinds of networks, right? So we often characterize this using their shear modulus. So shear modulus, for those of you I'm not assuming anybody knows anything, so I apologize if something is too simplistic, right? So basically, there are multiple ways of measuring shear modulus. One, the simplest way is you put your material inside a rheometer, right? And then you shear it, right? And you measure the resistance of this material to shearing, right? And if something is more solid-like, it will have a finite shear modulus. If something is more liquid-like, it will have a zero shear modulus, right? So for biological systems, things are often very small. So we don't do this rheology thing. That is more, we call it macrorheology. We do something which is known as microrheology, where you sort of embed beads into your sample and you look at how these beads are moving. And from the movement of the beads, the mean square displacement of the beads, for example, inside your sample, you can measure, again, what is the modulus of this material, right? And you can look at like 1-point microrheology, where you're looking at, thermal motion of individual beads or even active motion of individual beads. Sometimes you do 2-point microrheology, where you're looking at correlated motions of beads, etc. So once again, that gives you an idea of what is the sort of, shear mechanics or shear modulus of the system. So here I'm showing you an image of what this looks like for in vitro actin networks from a paper by Margaret Gardell, one of the organizers of this school. And this was from her PhD. And so again, 2 decades ago. So she was looking at in vitro actin networks. So she took this actin, purified actin from cells and then sort of made networks of it in vitro, right? So not inside the cell, but outside, right? In a controlled sort of manner, and then studied the modulus, the shear modulus, in the paper, she calls it elastic modulus, as a function of how much actin is there, so the concentration of actin, and what is the density of crosslinks? Because remember, I told you that these polymers can be cross-linked, into networks using crosslinks, right? And as you can see, she finds that there is a regime where, you have shear modulus, which is of the order of which is 100 pascals, right? Once again, even though the Young's modulus is gigapascals, when they form networks, that modulus will come down quite a bit, right? Because now you have a network of these filaments. That's going to be of the order of, 100 pascals or so on. So, and then as you decrease either the density of crosslinks or the actin concentration, you can see that the system can behave very fluid-like, right? So it can be either more elastic or more viscous or fluid-like, depending on how much filamentous material is there, what is the density of filaments, what is the density of crosslinks, and so on, right? So this is how people measure things in experiments. So if you're doing a theoretical calculation or a computational simulation, you can ask, well, how am I going to predict whether this network is rigid or not, right? And then, so first of all, is it rigid? Is it not rigid, right? Just that yes, no question, right? And how do we predict the value of this shear modulus for networks made up of different types of biopolymers, so of actin or microtubules or intermediate filaments, if you vary the type of biopolymers, or if you vary the concentration or density of these filaments, how is G going to differ with those kinds of things, right? So one approach that we use in theory and simulations to study this is known as rigidity percolation theory, right? So today's lecture, I'm going to mostly use sort of introducing you and discussing rigidity percolation theory, right? Questions so far? Yes. Can you speak a little bit more loudly? What do you mean by diffusion of what are you thinking about? So the thing is, so a couple of different things. So the regime where we are not talking about what happens when the actin is polymerizing and making filaments, so the monomers have to diffuse to the site and make the actin filaments. We are already in the regime where I'm talking about, we are already in the regime that the filaments have formed, their lengths are stable, so they are not undergoing polymerization, so these experiments were done on filaments whose lengths are stable. So they're not undergoing any more polymerization or depolymerization when these networks, are being studied. And the other thing is that we are also sort of in a limit where thermal effects are not that important, right? So essentially, for these kinds of properties, we are mainly focusing now on mechanics. and not on other things, right? But if you are actually looking at the formation of the, like network formation as you're polymerizing, and then network dissolution as you're depolymerizing, etc. There, both the rate of polymerization, depolymerization, as well as diffusion of stuff, although diffusion is not the more important thing, those things are all going to be important. But here we are looking at, filaments have already are some, stable lengths, and they have already formed networks kind of situation. And then looking at the mechanics of those networks, right? So not the whole dynamical process. Okay, so what is percolation? So how many of you know? Okay, I guess what are people's? Many of you know what is percolation, but when you hear the word percolation, what comes to your mind? Coffee grounds and coffee. Okay, so what about that? A little bit more. Yeah. Yeah, so how water flows through porous material is a very good example. I'm going to give you another example of percolation, which is a very different kind of example. So it's basically thinking of percolation, right? Let's say percolation of rumor in a cell phone network, right? So think of a town which has a lot of people, and people have their own, sub like networks or connections, right? And let's say you start a rumor at one edge of the town. What is the probability that the rumor is going to percolate through the whole thing and reach the other edge, right? So you can ask, how many people with cell phones have to be there, and what kind of connectivity should be there between these people for this rumor to propagate or percolate, right? So you can imagine that if there are not enough people or not enough connections between people, this rumor will not propagate. But then there will come sort of a critical number of either in terms of people or their connectivity where this rumor will propagate, right? So percolation is important, in many kind of systems. This is an example of connectivity percolation, where what matters is basically the connection or the connectivity between these people, right? So the type of percolation that's important when we think about mechanics, right, is rigidity percolation, right? So not just connectivity, but rigidity percolation. So what is that? So let's say I start with a network that's on a lattice. And here I have chosen a Kagome lattice, and I have diluted parts of the lattice. So I've removed some of the bonds uniformly, right? And only 45% of bonds are there. Now, if I try to sort of mechanically shear this lattice or load this lattice, it's not going to be able to bear this load. It will have a zero shear modulus, right? Because it's completely floppy, it's not rigid in any way, right? Then what I can do is I can increase the number of bonds that are there in this lattice and make it 50%. Okay, more bonds, but still, if I try to shear it, it will not be able to resist this shear. I can increase it even more, 55%. Now things are connected, right? But systems achieve connectivity percolation with a smaller fraction of bonds than rigidity. So although you start seeing connectivity emerge, right, it will not have a finite shear modulus yet. But then what will happen if I increase it even more, and now I go to 60%, right? So at some point, this network went through a mechanical phase transition, right? As I'm changing the fraction of bonds, it went from having a zero shear modulus to a finite shear modulus. And this phase transition, it's a phase transition because I'm changing this thing like the amount of bonds, the number of bonds, the amount of filamentous material. I'm tuning that by a small amount, and a characteristic property like the rigidity goes from zero to finite, right? So that's a characteristic of a phase transition. Yes. So rigidity transition, this is also a percolation transition, but the previous one was just a connectivity percolation, and something may be connected but may not be rigid. Rigidity percolation, like a system which percolates rigidity, will already have gone through the connectivity percolation transition, but it needs some additional more bonds, more constraints, right? So there is some system size. So the thing is, in the thermodynamic limit, there should not be any system size dependence. Thermodynamic limit is like infinite system size, right? Then the percolation threshold is fixed. It's purely a property of the geometry of the system and the dimension. But real systems are finite systems, right? So there is always some finite size scaling, and there is a small change in the percolation threshold with system size. Yeah, so people do study finite size scaling of these kinds of things. So the thing is, like if I did, and I will show you a mean field theory calculation. So when we do a mean field theory calculation for these kinds of things, we are assuming thermodynamic limit. But when you're doing computer simulations, you will do, some finite size, even with periodic boundary conditions. So for computer simulations, the value you get for the percolation threshold may not be exactly the same as what you get from, let's say, your mean field theory. So then what people do is they do their simulations at different sizes of the systems and look at what that scaling looks like, right? Yes. It is, yes. So the order of the phase transition, this is something I'm going to talk about in next class, in terms of first order versus second order, what's going on, et cetera, and different types of phase transitions in biological systems. Yes. But so maybe delayed gratification, wait until tomorrow. Yes. Yeah, you and then you. So what we will see is that the shape of the curve, the modulus as a function of the number of bonds, that will change based on the stiffness, but the location of the transition does not depend on the mechanical properties of the bonds. Yeah, so there is a reason. Okay, I can tell you the reason now. So the thing is, we were studying biopolymer networks, right? So normally, other lattices that will come to your mind before a Kagome lattice, first is a square lattice, then a triangular lattice. Why not a square lattice? We will see why not a square lattice. Essentially, a square lattice will always be floppy to shear because of what a square lattice is like. And we will see that today. Why not a triangular lattice? A triangular lattice could also work well, right? By the way, this lattice has both, the filaments here have both stretching and bending. So triangular lattice, when a triangular lattice is fully ordered, at any cross link, you will have 3 crossing filaments because your coordination number is 6. But in biopolymer networks, at any cross link, you will have 2 crossing filaments. So you will have to dilute a triangular lattice a whole lot to come down to this 2 crossing filaments or less situation. And that will interfere with some of the other things, mappings with biopolymer networks. Yes. Yeah, so people do, calculate dispersion relationships. So towards the end of today's discussion, I will talk a little bit about the dynamical matrix, right? And yes, so people do calculate, look at different modes, and dispersion relationships for these kinds of things. But we'll have to do a little bit more calculations. We'll have to do some math before we get to that point. Were there any questions on this side that I missed? Yes. So that's coming, right? So that's coming. That's coming. We will first see sort of a more phenomenological way of doing this, and then a little bit more, Okay, if you increase the number of bonds even more, it will stay rigid, of course, Okay. So now your question, right? So to simplify the situation, we are going to think about not semi-flexible networks where there is both stretching and bending, but only networks made up of only springs, right? So there is only stretching. We are not worrying about bending. So in these kinds of networks, we can make a prediction about whether this network is rigid or not rigid just from simple constraint counting arguments, which goes by, which is from Maxwell. and it's known as Maxwell's criterion, right? So this is in this paper from before any of us were here. But this building was, yeah, no, yeah. Okay, of course. So think of a lattice which has n nodes, right? So n nodes, and the lattice is in d dimension. You can think of 2 or 3 dimension, if that helps in sort of visualizing things. The coordination number is z, right? So coordination number is what is the number of nearest neighbors for any node, right? And the probability that a bond in this lattice is occupied or not is p, right? So the number of degrees of freedom in such a lattice is the total number of nodes plus the, not plus, times the dimension, right? So it's n times d, right? If there were no, if you're not thinking about constraints or anything. And then the total number of constraints is, n times the coordination number, right? So how many nearest neighbors does each thing have? And times whether this, bond, so let's think of this, right? So in this case, the coordination number z means 1, 2, 3, 4, 5, 6. There are 6 of them, right? And all 6 bonds are occupied. So P is whether, like what fraction of these bonds are occupied, right? Why do I divide by 2? It's because a pair of nodes share a bond. So if I do not divide by 2, I'm going to double count things, right? When I'm counting constraints. And in this case, I'm considering a spring network or a central force network, and I'm only considering nearest neighbor bonds, right? So only 2-body kind of forces or constraints, right? So the total number of degrees of freedom is n times d, and the total number of constraints is n by 2 times z times p. Do you all agree with this? Right? So what Maxwell's criterion tells us is that just at the cusp of rigidity, these 2 things should balance each other, right? If it's not rigid, then the number of degrees of freedom is going to be slightly more than the number of constraints. And if it's rigid, then the number of constraints is going to be slightly more than the number of degrees of freedom. But just at the edge of that, they are going to balance each other. Right? So if we balance each other, and that gives us this rigidity percolation threshold, right? What is the threshold for this? If you equate these 2 things, right, the n and the n will cancel, and then your P is going to be 2D divided by Z, right? So PC, the percolation threshold for central force networks, like spring networks, central force meaning you have 2-body interactions like, spring, Hookean spring interactions. And you can also define basically what is ZC. ZC is basically what is the value of this so that this PC is going to be 1. So that's basically 2 times D, right? So this is known as isostatic coordination, right? So this is the coordination at which you are basically just at the cusp of, above this, you're going to be rigid. Below this, you're not going to be rigid, right? This is for P equals 1. Yeah, ZC is decided by P equals 1. And so in 2 dimension, right, ZC is 4, right? So for a square lattice, ZC is 4, right? For a triangular lattice, what is ZC? Oh, ZC is 4 for everything. In 2 dimensions, ZC is 4. Sorry. PC for a square lattice is what? 1. PC for a triangular lattice is what? What is Z for a triangular lattice? 6. So for PC for a triangular lattice is 2/3. So for a square lattice, all of the bonds have to be present for the lattice, to have like, and even then, P equals 1 is not like, you will never get to P greater than PC, right? So a square lattice, you can imagine if you made a square lattice, if you shear it like this, it's not going to resist it, right? For a triangular lattice, because it's 2/3, if you are above this 2/3 threshold, then, you will have rigidity in your lattice. Yes. Why do we have a factor of half here? Okay, let's see if any of your classmates wants to clarify or add there. So why do you guys think somebody wants to volunteer why we have a factor of half here? Yeah, so these two, you're counting this bond for this, but also for this, right? So because each bond, each constraint is being, if you don't multiply by half, you're counting each constraint twice because it is attached to this node, but also this node, right? So does that Maxwell constraint counting? So for any kind of central force network, doesn't have to be, I'm only showing you examples in 2D. In 3D, also the same things will hold. It's just that, your dimension is now 3, right? So if you did simple cubic or FCC or BCC, again, this constraint this kind of thing should hold, provided you have a central force network, like a spring network, right? Now, for a Kagome lattice, it's a 2D system, D is 2, right? And number of, Z is also, this thing is also 4, like a square lattice. For a Kagome lattice, with only springs, it has the same issues as a square lattice. However, once you add bending rigidity, it will increase the number of constraints. And so a Kagome lattice will become rigid once you have added bending rigidity. But without bending, if you only had springs, it will have the same kind of problems as a square lattice, right? But square lattice has a very special pathology that even if you add bending, it still has this issue that, we will still have this, will not get this finite shear modulus. So you'll never see people using square lattice unless they have added some kind of a different next nearest neighbor or different kind, like a other kind of thing. So, okay, so then again, these networks that I was showing you earlier, all of these networks, if you look at these, will have a coordination number less than ZC in 2D, less than, and if you're thinking of 3D networks, which sometimes they are, right? Especially if you're thinking of collagen, ECM is often a 3D network, right? So the connectivity is definitely, in 3D, it has to be even more. It has to be 6, and it's definitely less than that. This is also, it's less than 4, So then you can ask, how can these networks be rigid, right? I showed you that for these networks to be rigid, ZC has to be Z has to be greater than ZC, right? The coordination. But these coordination numbers are all less than, 4, right? And in this case, less than 6. This is a 3D thing. So, and, experimentally, people measured the rigidity and shear modulus of these networks and find a finite value. So what are we missing? Anybody who is not a professor. So Leo is not allowed to say anything, and Angela or Marcus also no. So the rest of you, yes. Yes, indeed. So we are only thinking about spring networks here, but these filamentous networks are all semi-flexible. Remember, I told you. So then there is bending, and there will be additional constraints from the bending, right? So so those additional constraints are neglected in this constraint counting that I just did. If you had included those constraints, then, you will have rigidity, right?
But the angles, so let's say, even without bending,
yeah,
there's no rigidity for the angle.
There is no rigidity for the angles because even if you had angular constraints, right, those will also be a type of bending, a type of, additional constraint. Yeah.
But they're distinct from bending the bond.
Yeah. So we are only thinking of filament bending, which is bending for bonds, which initially make 180-degree angles. But you could also have angular springs, and it depends on types of crosslinkers. So there are some types of crosslinkers, like filament, for example, in case of actin, which will sort of like to crosslink actin at like 90-degree angles, and any change from that angle will be penalized, right? So that's another type of bending, where there will be energy penalty for changing angles between. So for these kinds of things, there are different crosslinkers. Some crosslinkers crosslink things in a very sort of floppy way, so the change in angle is not penalized. And for some crosslinkers, the changing in angle is going to be penalized. All right. Indeed, you were right. This is because of bending rigidity, which makes the semi-flexible, and constraints, additional constraints, which we will discuss tomorrow. But I just want to at least tell you what we do with these kinds of semi-flexible networks, and we will get into the details tomorrow, right? So let's say often, if you want to do theory with these kinds of networks, so you can construct these networks either in an off-lattice manner, right? Like a disordered network of filaments in a Mikado model kind of thing, or you can build these networks on a lattice and then get disorder by diluting the lattice by removing bonds, right? If you want to do analytical calculations, the lattice approach is the approach we usually follow, right? So here, for example, this one is, I'm showing you things on a triangular lattice. Again, another lattice that we sometimes use. And so, we basically will write down the deformation energy for these kinds of networks. And, before we do large deformations, we want to do small deformations, like so that you're in the linear elastic regime, everything is changing, very slowly, right, or very little. So in this linear elastic regime, or very small deformation regime, the elastic energy of these filaments, you can separate it out into two parts. It's sum of two parts. There is a stretching energy, which is the spring energy, and then there is a filament bending energy, right? So stretching energy, basically, Uij is basically the deformation of a bond, right? How much it has deformed, the Ij bond. Rij is the unit vector, right? The direction of the unit vector initially for the undeformed lattice, right? And this Pij is whether this bond is there or not, because we will consider contributions to stretching only if the bond is there. Otherwise, there is no contribution. And then you can sum over all of the bonds, right? And then you will get, a stretching energy. This mu is the sort of stretching rigidity and so on. You can also have a bending rigidity, right? And there, basically, what we are looking at is, so theta ij is this angle between pairs of bonds, right? So initially, so filament bending is only considered for, pairs of bonds which were initially making a 180-degree angle. This is what Leo was asking, right? So there can be multiple types of bending. You can have filament bending where, you're looking at only series of bonds which initially make, 180-degree angles are like this. And then these bonds, there can also be, you can have sort of crosslinks which constrain the angle between crossing filaments. But in the simplified models, the first order models, we say that, okay, our crosslinks are floppy, so they will not constrain these angles, right? So the only bending rigidity is coming from filament bending. And why are there two of these P's? Because for this bending rigidity to have a non-zero value, both of these bonds have to be there, right? So the bending rigidity sort of resides in a hinge, right? It doesn't reside on individual bonds, but it resides on bond pairs. It's kind of a three-body interaction because you have three nodes which are participating in this, right? And the total energy is going to be the sum of all of the stretching energy over all of the thing and the bending energy over all of these guys, right? And then one could do an effective medium theory, which is a type of mean field theory, or one could sort of minimize this energy. So you can apply a shear or a compression or an extension, whatever type of, modulus you want to calculate, etc. And you can sort of, apply that small deformation, right? And let the system relax, minimize its energy, find what is this, energy minimum, and then you can calculate your moduli from that energy minimum, right? Yes. If your system is in equilibrium, you will get a minimum. Yes. It might be a very, if you're doing a simulation, often we do a conjugate gradient minimization just because it can be a very corrugated landscape, right? So. Yeah, but you will have a global minimum usually, or you'll, yeah. But it's not going to be just, if you're asking, is it going to be a parabolic system? Not necessarily. Yeah, so that's not necessarily true because it's a multidimensional system with so many everything, right? So yes. Question and then question. No, so the bending, think of a filament as a contiguous series of bonds, which initially made 180-degree angles. And every time you have a filament, you have a bond pair, you have a hinge. So the overall bending energy is going to be the sum of the bending energy of all of these hinges at all of these bond pairs at each filament. And then you add over all of the filaments, all of the bending. Yes.
A related question. If you maybe like reiterate or possibly draw a picture of one of these vertices where the bond is floppy and where there's not this same kind of three vertex bending energy.
So when I say the floppy part, when I was talking about that, that was not for, so these bending energies are always there. When I said floppy, what I meant is, so I guess I was going to go into that a little bit later, but let me talk about this, the network construction. So how do we construct this network? What do we mean, right? So initially, let's say you're in 2D, you throw down infinitely long filaments in a lattice kind of way, right? It could be a triangular lattice, a square lattice, a Kagome lattice. Think of your favorite lattice. So you have filaments arranged like this, in a triangular lattice, then like this, right? Then we say that all these filaments are infinitely long, right? Then we say that, well, before we do anything, we want a broad distribution of filament lengths. So then we are going to take, and you can think of these filaments as a series of bonds, right? So you can start diluting bonds from the lattice in a sort of uniform manner, right? In a random uniform manner or a Gaussian, either a uniform distribution or a Gaussian distribution, but do it sort of, randomly like this, right? So now you will have a broad distribution of filament lengths, right? Because sometimes you will have 2 bonds, then maybe 10 bonds together, maybe 20 bonds, maybe 5 bonds, right? So filaments of different lengths, right? So now we say that every time 2 of these filaments go on top of each other, there is a crosslink. So that's an assumption of the model. Anytime you have 2 filaments that are crossing, there is a crosslink. And what these crosslinks do is they will, prevent these filaments from sliding on top of each other like this, right? So this is a hard constraint. No matter how much energy you give it, it will not allow it. But the first type of model one could think of is these kinds of change in the angle between crossing filaments. This is not constrained. So basically the crosslinker that's here, it's so floppy that it's not changing this angle. However, like Leo was asking me, for actin networks at least, there are different types. There are 23 or more, at least 23 different types of crosslinkers. Some of these crosslinkers will not constrain this angle, and the filaments can do this. They can rotate freely. And some of the crosslinkers will constrain this angle, so that if you change this angle, then there is going to be an energy cost. So when I said floppy, I meant for this angular crosslinkers. However, filaments always will have a bending rigidity. If you're thinking of actin or collagen or intermediate filaments or microtubules, the ratio of the relative bending to stretching cost changes. It's always bending is always easier, like it's always easier to bend than stretch, but whether it's only a little bit easier or it's much easier depends on the type of filament, right? So the ratio, the scale, like non-dimensional ratio of bending to stretching could be 0.1 or 10 to the minus 4. It depends on the type of filament. But it's a finite number, it's not 0, right? So, because these are semi-flexible filaments. Yes?
Your lattice up in a sorted way like this, do you expect your energy landscape to be glassy, like construct at all scale?
I don't use the word glassy loosely because I think glass means a very specific thing, but it could be very disordered. Yeah. So if you're asking, is it a disordered system? It's a very disordered system. It's a very disordered network. So for a system to be glassy, it has to have a lot of other properties as well. So, so it could be a very disordered system. Whether it's glassy or not will depend on a number of other things, right? So, yes. Yeah. Yes, you're a bit confused? Yeah. 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 Well, so the thing is the bond, the bond is not a body. When I say three body, I mean three nodes, right? So I, J, and K. So it's so central force interactions are two node interactions, right? So I and J. This is a three node interaction, I, J, and K. So when we do percolation theory, two node interactions and three node interactions have very different types of contributions. When we do those calculations, you will see. But when I say three body, I mean that there are three lattice points or lattice nodes that are connected, right? Not the constraints themselves, right? You had a question, and Leo has a question. Yes.
Yeah, so I think what's a little bit confusing, you compare, at least to me, compared to what you're saying.
Yeah.
So, in the picture, you're drawing a node, which is an intersection, a cross-link between.
So node is, you can think of the node as a crosslink. It's just, yeah,
Right.
yeah.
But when you're describing, you're also saying there's bending energy of a bond away from the vertex.
No. So bond, yeah, bond is a bunch of segments. Bending energy is not residing in a bond. Bending energy will reside in bond pairs, which are sort of forming a hinge in this
No,
model.
but do they have to? So the energy that's lit there and drawn there.
Yeah.
It's always about bending.
It's basically how this angle is changing. So initially it was this, and now it has become this.
Right, but only around vertex. But you could also think about bending the bond itself, like
So if you're thinking of bending a bond itself, then you will need a midpoint of the bond, etc. But we don't have a midpoint of the bond or any other discretization of the bond itself, right?
Well, I mean, you can bend it into a knot, like you just.
So that is that this model doesn't allow it. Yeah, this model doesn't allow that. Yeah.
The other question. These are zeros and ones. They're occupied or not occupied.
Yes. So yes. So you're absolutely right. So for example, in this kind of model, if I wanted buckling within a segment, I cannot do that because it's not there. I will have to at least introduce a midpoint if I wanted buckling, right? So that's a model assumption, and that I didn't state. Yeah, so that is an assumption of the model. Yeah. How am I doing on time? Yes, question. When they are initially in the straight line. So basically, like I was saying, there are two types of bending. One is filament bending, and the other is if you have angular crosslinkers between crossing filaments. We are assuming that the crosslinkers between crossing filaments do not constrain angles. So the only type of bending is the filament bending, where initially the filaments were straight, right? So their rest state is that they are kind of straight. Okay, so I guess I discussed a little bit of that, how to construct this kind of a network. This is something that, I loosely said in words, but the way we are diluting bonds, if I wanted to write down what is the probability distribution of spring constants, right, of bonds, and what is the probability distribution of bending constants of bonds, how do I write that down? So mathematically, this is how we write it down, right? So, basically, or maybe we'll come back to this later. Some of these things will become zeros. Maybe I come back to this point later once I have actually discussed the calculation. So often, if we want to calculate what is the shear modulus of the system, right? Is it going to have a resistance to shear or not, right? So the theory that we often do is known as an effective medium theory, and it is a type of mean field theory. So the main principles of the theory is that you take a disordered lattice, so a whole bunch of bonds are missing. This is a very skeletonized picture of a very small part. And let's say the stretching energy of the filaments is alpha, the bending energy of the filaments is kappa. So what effective medium theory does is it says that imagine an effective network that is not disordered, that is completely ordered, where all of the bonds are there, right? And this effective network has the same overall mechanical response, right? Overall mean field mechanical response as this disordered network. So if you put this thing inside a rheometer and this thing inside a rheometer, and you measure the shear modulus, your rheometer will measure the same shear modulus, even though this has far fewer bonds than this. So then it asks, for such a completely ordered network, what does individual alpha m's and kappa m's have to be, right? So that the mechanical rigidity is the same for both of these things, right? So an effective medium calculation lets you relate this alpha and kappa to some effective medium alpha m and kappa m. Yes. Can you speak a little loudly? Is it?
Like, if I need one of these disordered ones, then I always find unordered ones.
So things can get complicated and messy. So for example, I'm thinking of a completely isotropic system. Now, if you have an anisotropic system, then you can ask, well, in an anisotropic system, you usually need multiple independent elastic constants. Does it make sense to think of only, one alpha m and one kappa m if you, let's say, have, or would you will need, like 5 or 9 or whatever number of, elastic constants independent, depending on your systems. So you have to ask those kinds of questions, right? Yes, you can calculate it, but would it make sense for a system like that? But you can still calculate it as an instructive thing that, okay, what would it look like if I calculated something like that, right? So, yes.
Isotropic, but even if these have the same shear modulus, presumably the network on right with fracture and much lower strain, since it is more densely cross-linked, or is that not the right way to think?
Yeah, so fracture is a very different thing. And there, of course, the actual configuration matters. So you are absolutely right. So here, in the mean field sense, we are only thinking about, the effective shear modulus kind of thing, right? And the other thing that when I wrote down the energy, remember, I wrote down everything in small deformation limits. I'm only focusing on the linear response regime, right? For this part, at least. When you have nonlinearities, and fracture is a very nonlinear, phenomenon, things will change. I am hoping to talk about fracture at least not next tomorrow, but like Friday, the last day, I hopefully will talk about fracture. Yes. Yes. So for semi-flexible networks, for isotropic networks, again, these things match up for semi-flexible networks, etc. For anisotropic networks, things are messy and not as clean. And then I should also mention that people who study granular materials often construct packing-derived networks, which are not networks like I drew them, right, on a lattice like this. If you construct packing-derived networks from granular materials, there the shear modulus and the bulk modulus percolation, like the thresholds for rigidity, may not necessarily match up, do not necessarily match up. But for biopolymer networks like the ones we are thinking of here, they will match up. Yes. No, this is just a skeletonized thing showing you what is the meaning of effective medium theory. So I would say don't take this network too seriously. One of the things that we will calculate in the effective medium theory is, where is the rigidity percolation threshold? Does it agree with the constraint counting argument that we found from the Maxwell criterion, right? But the constraint counting argument only tells you whether something is rigid or not. What the effective medium theory does, it tells you whether something is rigid or not, but it also gives you what the shape of G as a function of P looks like, right? Given a certain P, what is G? And then you can do it for a series of P's, and then at P equals PC, G is going to go to 0, and then below that it will stay 0. But above PC, it will have some shape, and we'll find out what that is. All right. I have 20 minutes. I will see how much I can do in that, and we will continue, right? That's the good thing about these lectures, is that, I'm not bound. I don't have to rush. I will not rush. I think it's better you ask questions and I care, like, you understand things rather than me rushing. Okay, so we will do this thing for a simpler system, which is, I want to show you this calculation first for only springs. So, okay, so effective medium theory for central force or spring networks. This is a very famous paper. So, these days, all of the fancy papers are in Nature and Science and PNAS and whatnot. There used to be a time when some of the best papers that we worked through were PRAs and PRBs, right? So, and this is a really, really good paper. And I really encourage all of you to read this paper. It's one of my favorite papers ever. It's by Shichao Feng, Mike Thorpe, and Garbozi's first name I forgot. It's a PRB from 1985. It's not a very long paper. And it's very, very, you can easily work through this paper. It's a really nice paper. So I will highly recommend this, right? So this paper is the first paper on effective medium theory for, spring networks, right? And then other people have made more complex versions of this. I, for example, did this for semi-flexible networks where there is also filament bending. But again, the gist is the following. You have a spring network that's disordered, and the bare spring elasticity of the springs is alpha. We ask, if we had a completely ordered network where all the bonds are present, what would this alpha m have to be so that both of these networks will have the same overall mechanical response, right? So that's what this calculation will tell us. So we will focus on one connection, right? Let's say focus on the connection between this point and this point, right? Now, each of these bonds have a spring constant alpha m. However, if you ask, what is the elasticity of this connection, not just this connection, of the connection, overall connection between these 2 points? Is it alpha m? No, it's going to be greater than alpha m. My claim is it's going to be greater than alpha m. Why? So this bond is alpha m, but the elasticity between these 2 points is greater than alpha m. Yes. Yes, yes, you're exactly right. Because these 2 points are not just connected by this bond, but they are part of a network, and they're also connected by other springs, right? So the overall elasticity, stretchiness between this node and this node is going to be greater than alpha m. We write it as alpha m divided by some constant A star, which we can calculate, and we will calculate either today or tomorrow, depending on how much time we have. This A star comes from the geometry of the lattice, right? Does it make sense why this is greater than alpha m, right? It's not exactly alpha m. Because again, even if you think about this, even if you remove this bond, there will still be some elasticity for these 2 nodes because they are part of a network, right? So now what we do is we put this network under, strain uniformly, right, overall. So, and we take this ordered network. We start with the completely ordered network. We put this completely ordered network under uniform strain so that every bond is stretched by an amount delta um, let's say, right? So every bond in my network is stretched by this amount. Then what I do, I replace one of the bonds by a different bond, which has a different spring constant, not alpha m, but a spring constant alpha, right? So everything else is the same, but I replaced one bond by a different bond, which has a different spring constant, and this spring constant is alpha. So now if I want to find out what is the effective elasticity between these 2 points, it is alpha m by A star, which was the initial thing, but now I'm replacing alpha m by alpha. So I subtract out alpha m and I add alpha, right? So now this is the effective elasticity between these 2 points when I have replaced this bond by a different bond of the spring constant. Are you all with me? Yes.
Alpha the same, alpha is the network, or is it arbitrary?
Forget about the disordered network or anything. This is just a new calculation. Think of this as a new calculation. We will get to the disordered network, but this is a calculation where you start with a completely ordered network where all the spring constants are alpha m. And I have removed one of the springs, alpha m springs, and replaced it by a spring of spring constant alpha. Yes. Sorry. So here, this part, well, so the thing is, this is the overall this thing, and then I just replaced one bond by another bond, right? So it's just the, I'm just focusing on that particular bond here, right? A star is less than 1. Yeah, and we do a dynamical matrix calculation to get the A star. That I may not have time to do today, which I might do next class. We'll see. May or may not be. Yeah, we are not really saying anything. So then one of the questions you can ask is, so now because you have replaced this alpha m by alpha, there is going to be some additional deformation, right? Everything was initially under a strain delta um, but now one of the bonds is different. So then this bond will have some extra deformation, right? What is that extra deformation? We don't really know, right? So how do we calculate this extra deformation? So then Feng and Thorpe and Garbozzi proposed the following way of calculating this extra deformation. So do you guys all agree that there is going to be an extra deformation now in addition to the uniform deformation that everything has? Because this bond now has a different spring constant, right? So either depending on, whether it's larger or smaller than the alpha m spring constant, right? There is either going to be an extra compression or an extra stretching of this particular bond. We want to find out what this is. We don't know, right? So how do we find this out? So first we think of so a virtual, this is a virtual force. So what force we would have had to apply to this bond to bring it back to the situation, right? Before there was this extra deformation. That virtual force would have to be basically this alpha m minus alpha, right? Because this is the difference in the spring constants times the uniform thing that we had applied, right? So the way they suggest we calculate this is we take this virtual force. What is the force that's necessary to bring the springs to their original position? Original position meaning position before you replace this bond, right? Then you take this force and you apply this force on this bond, right? And then, this du, the extra displacement, is this force divided by the overall spring constant of this bond, right? So this is a way of getting this fluctuation in the displacement or change in this displacement because we have replaced the bond. All right. Okay. I can replace the F here. So this is my extra displacement that this bond had, right? Because I replaced, alpha m bond by alpha, this is the extra displacement it has. So then you can say that, okay, what if I didn't just replace one bond? I made a disordered network where I did a whole bunch of different things. So that's the next step. We'll come back to this if you have questions. So for a disordered network, some bonds are there, some bonds are not there, right? So there, the situation is sort of over a larger scale. Some bonds have a spring constant alpha, some bonds have a spring constant 0. So there, what we do is we average this fluctuation over the entire system, and the fluctuations will go, like when averaged over the entire system, it's going to go to 0. So you take something like this, you multiply it with this kind of a disorder distribution, right? And then you average over everything, and then this goes to 0. So basically, what this distribution means that the probability that a bond is there and has a spring constant alpha is 1. Is P, and the probability that there is no bond, right, is 1 minus P here. right? So if we say that the fraction of bonds that are there are P, then we write it as P times, the probability that this bond has a spring constant alpha. And then you have 1 minus P, and then, the bond has a spring constant 0, right? So alpha prime minus 0. So this is how we write down the distribution. Yes. Yeah, it's basically we are doing a mean field calculation. So we are saying if you average these small fluctuations over the entire system, they will go to 0, right? So. And if I plug in this distribution, the probability distribution, this one in this integral, then we end up with the simple situation, right? Alpha m by alpha is P minus A star by 1 minus A star. And again, A star was the A star that was in the denominator, right? Sorry, this is a typo. I was like, where is my A star here? This is not alpha prime, this is an A star here. So to use this here, what you will do is this is an A star. You will write this alpha and this alpha, these guys as alpha primes. You will substitute this distribution, do the integral, and then you get something like this, right? Okay. Yes. Yes. So we start with one modified bond, and I show you what is the extra displacement for one modified bond. Then I say that, well, if you had multiplied a whole distribution of bonds in the network, but the way you had modified it is that instead of replacing them by a different bond of spring constant alpha prime, it's as if, or alpha, you are basically just replacing them by nothing, right? You're replacing them by zeros. Then what would things look like? But you could also do a calculation where half of your springs are alpha and half of your springs are beta by following the same, or a fraction of springs are alpha and a fraction are beta by following the same rule, right? So this is not something we will have time to do, and it's a good thing that we can do it on the board. I will do it tomorrow. So then you can ask, what the heck is this alpha star, right? Which is coming in, because it's P minus A star by 1 minus A star. So if you look at this quantity, essentially at P greater than A star, we will have a finite alpha m. At P less than A star, negative just doesn't mean anything. It basically means that it's, not rigid anymore, right? So this relationship is only valid when P is greater than A star. And A star is basically my percolation threshold. Whatever value I get for A star would give me my percolation threshold. So how do we calculate A star? What is A star? These are things we are going to do in next class. So it's a geometry constant which relates the geometry of the lattice to its rigidity, right? Because again, remember, why do we end up with an A star? It's because the elasticity between 2 points depend not only on the direct connection between the 2 points, but on the overall geometry topology of the lattice, right? So this A star will depend on that. And we calculate A star using the dynamical matrix. And we will discuss all of this in detail in next class. What is a dynamical matrix? How many of you have seen a dynamical matrix in a solid state class before? 1, 2. Okay, some of you have seen a dynamical matrix. If let's see, what do you think of? How would you describe a dynamical matrix to your friends that you think they will understand? If somebody has never seen it before. This one is an inverse. Yeah. So how will you describe a dynamical matrix to your friends? How will you describe a dynamical matrix to a stranger you met at a party? You only knew that that person has had a physics undergraduate training. Yeah, so that's one way. So normally, you can think of it, it's a matrix which has a lot of components. These components are related to the second derivative of the energy, like she's saying, or it's the first derivative of the force. So it's like components which are all force constants, right? But because your system is a network of springs, right? It's not just a spring, a 1D spring, right? So you need all of these different components. So the components of the dynamical matrix, again, these will correspond to, if you think of different wave numbers, I guess we'll go into wave numbers, etc. in next class. Let's not complicate it more, but it's a generalization of, if you had a spring, an individual spring, you will have a spring constant. If you have a network of springs, now you have a dynamical matrix, and the components are related to that, right? So that's how I will describe a dynamical matrix to a physics undergrad at a party, right? So.
But maybe you can comment on why
How this thing is not, so this particular thing is 1 in this case.
no, I mean, usually a matrix, a normal matrix, like times its inverse.
Yeah, this one, in this case, A star is going to be 1. That's how we end up with the 2/3. But if you also had bending rigidity, etc., then it would not be a unit vector, 1. So in this case, this is 1. If you, yeah, it's in 2D, right? And if you take a trace of this, then it will give you a 2, 1 plus 1, 2. And then you end up with a 2/3, which is how you get a percolation threshold of 2/3 for a central force network. So it agrees with your constraint counting, but I will actually explicitly do this calculation, show you what this dynamical matrix looks like for a spring network. Well, even without doing this calculation, you know that this is going to be a 1. And then, because it's a 2x2 thing, right? The trace is 1 plus 1. So then it's like, it's 1, 1, 0, 0, right? So 1 plus 1 will give you a 2. So then you will get a 2/3 here. So for this calculation, it's simple. You can see it's 2/3. But I will actually show you how to, how do you write down the dynamical matrix? What does it look like for a spring network? Because when you have filament bending, then you will have to modify things, and then it's not going to be a simple 2/3. It's going to be more complicated, right? So next class, we will go through the dynamical matrix calculation for this thing, and then we will sort of generalize it to, semi-flexible networks, etc., right? The semi-flexible network part, I will do some things by hand, but I may not do the whole thing I don't know, should I show a Mathematica notebook? Well, we'll see, I'll decide something. But does it somewhat make sense what's going on, right? So I think we are at the end of time. Yes. More questions? Yes. Should we be surprised? No, if my calculation is correct, they should give me the same answer. So I think it's a good thing. We didn't so the thing is, this basically just tells you that the percolation threshold is a property of the dimension and the topology or geometry of the network. It does not depend on the mechanical properties of the filaments, etc. That's a very important statement. The mechanical properties are important. If you want to find out what is the actual rigidity, yes, the mechanics will inform that. What is the shape of the curve? Mechanics will inform that. But the threshold where this phase transition happens, where it goes from non-zero to 0, at least in this small deformation in this regime, it does not depend on the actual spring constant of the thing. So this is a very powerful result, that it will only depend on the geometry. Yeah, so we will discuss this again in more detail. So the mean field assumptions affect other kinds of things, not the percolation threshold, right? So the mean field assumptions, we overestimate certain things. We overestimate the G. If I calculate, let's say if I calculate scaling relationships, right? The scaling relationships you get from mean field and from simulations for the spring networks are still more or less in agreement. But if I did a semi-flexible network, then the exponents are slightly different from each other. They're not exactly the same. So the whole thing about mean field is that certain properties, even though it's a mean field, like a mean field calculation is a very kind of averaging kind of calculation, but it is powerful because there are certain things it can predict very well. So the transition, it can predict very well. The exponents can differ a little bit. And what do the exponents mean? It means the approach to the transition, that is, there are, because we made some assumptions, right? That is not exact. I mean, or the mean field and the simulations, there will be some difference, right? But the transition itself, it describes, everything maps onto each other really well, right? But the approach to the transition, the exact value of the G, the assumptions will affect those things a little bit. And you can make comments on whether they will underestimate or overestimate those values, right? Thank you. Yeah. Oh, yes. Can you speak a little bit loudly so everyone can hear and I can hear? So here, so for example, so in that case, you will need to have like things like transient crosslinks, etc. So things like what happens when you have transient crosslinks, what happens when you have motors, what happens when you have composite networks, all of these things, I'm going to at least give you some, examples from experiments and theory that some I have done, some other people have done in the last lecture, right? So here we are thinking of permanently cross-linked networks, right? But those would be transient networks, where transient crosslinks, where crosslinks can go on and off and things can sort of reorganize and remodel, right? So, and there is some work, there is not a lot of work on that from a theory point of view, but there is some work. So thank you.